Alright, let's go ahead and get started with part 2 here, talking about adaptive immunity. Okay, we're talking about the major histocompatibility complex, the MHC antigen complex, and co-stimulation are usually necessary in order to activate the lymphocytes. Co-stimulation means that both of them are working together, and it involves both the cytokines and certain surface molecules. Okay, then there's some antigen-presenting cells that stimulate proliferation of helper Ts. Okay, and the helper Ts then stimulate the proliferation of the B or T effector cells. Okay. Alright, so we see how the movement goes. Alright, so we look at the dendritic cell first. Okay, the dendritic cell takes in extracellular antigens. Okay, and then they process this and display it on the cell surface. Okay, those antigens, once they're presented on the cell surface, then are processed and bound to an MHC, or major histocompatibility class 2 molecule. And then this presents the processed antigen to a T cell or a helper T cell. Then the CD4 attaches to the major histocompatibility complex class 2 molecule. Okay, and then you see that the B7 and CD28 provides co-stimulation of the helper T cell. Alright, and then this process goes on where the activator helper T cells produce interleukin 2 receptors and secretes interleukin 2. And this helps the binding between the interleukin 2 receptors. Interleukin 2 then stimulates the helper T to divide and differentiate. Okay, many divisions, which you will not see in the diagram, will increase the number of the helper T cells. Okay, the activated helper Ts will then give rise to memory T cells. Okay, and this will provide long-lasting immunity. And the activated helper T cells give rise to functional helper T cells that secrete cytokines. Okay, and the cytokines regulate the activities of certain immune cells, such as B cells, cytotoxic Ts, macrophages, and result in the elimination of the antigens. So let's check out the T-cell dependent antigens.
Antigens, such as bacteria or viruses, are usually ingested and degraded into small fragments by phagocytes, such as macrophages. The small fragments are then complexed with a special type of protein called a class 2 major histocompatibility complex protein, MHC, and transferred to the surface of the phagocytic cell. Helper T cells recognize an antigen when it is presented on a class 2 MHC because helper T cells have special recognition proteins on their surfaces called T cell receptors. This interaction activates the helper T cells. The activated helper T cell is now capable of stimulating B cells to become antibody-producing plasma cells.
Okay, so now let's check out what's coming up next. In the lecture, we're looking at the T cells, and we see how they're functioning along with antibodies. Alright, adaptive immunity. Okay, adaptive immunity, remember that is specific. Okay, certain targets. Okay, and what we're talking about is inhibition of lymphocytes. Okay, they can be inhibited. But let's talk about tolerance first. Tolerance is a suppression of the immune system's response to an antigen. Tolerance is produced by the deletion of the self-reactive cells and the prevention of lymphocyte activation. Suppressor T cells can also suppress the lymphocytes. Alright, so let's check out what's going on here. We're going to look at some of these things. Alright, so first we're going to look at an antigen binding to a B cell receptor. Okay. And this will stimulate the B cell. but most B cell activation requires helper T cells. Those helper T cells will attach to these B cells and will form a receptor-mediated endocytosis program that's going to come in and bring things into the cells. And then the major histocompatibility complex class 2 molecule presents processed antigen to the helper T cells. Okay, then the T cell receptors bind to MHC class 2s and antigen complex, and CD4 then binds to the MHC class 2 molecules. The helper T cell secretes interleukin 4 and other cytokines, which will stimulate the B cells. The activated B cells will then proliferate and differentiate. Many divisions, not shown, will increase the number of B cells. The activated B cells will then give rise to memory B cells. Those memory B cells provide long-lasting immunity throughout an individual's lifetime. Okay, will it last the entire lifetime? No, but we're talking about months, years, depending upon which type of cells we're talking about. And B cells have a certain lifespan, which we'll discuss later. Okay, the activated B cells give rise to plasma cells. And these plasma cells produce antibodies. Those antibodies will hang around for quite a while and be able to identify invading pathogens. The antibodies are part of the immune response that eliminates the antigens that come in. Antibody-mediated immunity. Okay, antibodies are proteins, and these proteins have variable regions on the antibody. Their constant regions will activate the complement system or bind to cells, but the variable region is what is used to identify invading pathogens. There's 5 classes of antibodies that exist: the IgG, the IgM, IgA, IgE, and IgD. Pay very, very close attention. There's going to be some that are going to be very prevalent and some that you won't see very much at all in the process. Okay, so check out. Look at our heavy chains and our light chains. Okay, it's composed of heavy and light chains. A heavy chain is a big part of the main part of the Y. Okay, and then basically you've got this accessory part that attaches to the top of the Y, and that's the light chain. Okay, then you have the complement binding sites on the bottom part of the heavy chain, base of the Y basically. And the site of the binding to macrophages, basophils, and mast cells also occurs in the constant regions of the heavy chain. Okay. A couple of classes of antibodies and their functions. Okay. The most prevalent antibody is going to be IgG. IgG is going to be the answer to most questions about antibodies, because IgG, which antibody does this, which antibody does that. IgG is going to be the one because it's the most prevalent in most cases. Okay, it activates the complement and it promotes phagocytosis, or cell eating, where it eats the bacteria cells that have invaded. Okay, and it can cross the placenta and provide immune protection to the fetus and the newborns. And it's responsible for Rh reactions. So this is actually what triggers the RhoGAM response. Okay, what you need that RhoGAM shot for if you have an incompatibility of blood types in Rh. Okay, IgM makes up between 5 to 10%. Okay, and it activates complement. It acts as an antigen binding receptor on the surface of B cells and is responsible for transfusion reactions in the ABO blood system. Okay, remember IgG would be part of the Rh. Okay, and it's often the first antibody produced in response to an antigen. Okay, whereas IgG happens a little later on. Okay, IgA. IgA is an antibody that is secreted in the saliva, tears, and mucous membranes, and it provides a little bit of protection on the body's surfaces, and it's found in the colostrum. Okay, remember that what the colostrum is? Okay, question number 1 for this lecture. What is a colostrum? What is it? Who knows? Right? And milk to provide immune response to the newborns. And IgE. IgE binds to mast cells and basophils. And and it stimulates the inflammatory response. Okay, IgD functions as an antigen binding receptor on B cells. Antigen-mediated immunity. Antibodies that affect the antigen in many different ways. Okay, it binds to the antigen and it interferes with antigen activity or binds the antigens together all together. Okay, binding to the antigen and interfering with antigen activity, or it binds the antigens together. Okay, holds the antigens together. That way, the complement system and the immune system can do the job by holding the antigens in place. It increases the phagocytosis by binding to antigens and macrophages. And it can activate the complement system through the classical pathway, not the alternative pathway, but the classical pathway. And it attaches to mast cells and basophils and causes the release of inflammatory chemicals when the antibody combines with the antigen. Okay, so here's what we got going on. So we're looking at, sometimes we've got the antigen binding. An antibody binds to an antigen and inactivates it. Okay, the antibody just completely shuts down the antigen that's causing problems. Then the antigen binds together, and the antibodies bind several antigens together. It activates the complement cascade, and the antigen binds to an antibody, and as a result, the antibody can activate the complement proteins, and once that happens, it causes inflammation, chemotaxis, and lysis. Okay, and then it initiates the release of inflammatory chemicals. An antibody that binds to a mast cell or basophil, and when an antigen binds to the antibody, it triggers a release of chemicals that can cause inflammation. And finally, it facilitates phagocytosis. Okay, eating those antigens by your macrophage cells. Okay, an antibody binds to an antigen and then to a macrophage, which phagocytizes the antibody and the antigen. Antibody-mediated immunity. This is caused by antibody production, and the primary response results from the first exposure to an antigen. And the B cells form plasma cells which produce antibodies and memory B cells. The secondary response results from exposure to an antigen after a primary response. Memory B cells then quickly form plasma cells and additional memory B cells. Okay. Primary immune response, as we see here. Okay. The primary immune response occurs when a B cell is first activated by an antigen and the B cell proliferates to form plasma cells and memory B cells. The plasma cells then produce antibodies. And then the secondary immune response occurs when another exposure to the same antigen causes the memory B cells to rapidly form plasma cells and additional memory B cells. The secondary immune response is faster and produces more antibodies than the primary immune response altogether. So we're going to see the formation or how a monoclonal antibody actually...
Monoclonal antibody preparations contain only one type of antibody, derived from a single cloned B cell. Consequently, they are highly specific for a single epitope and have applications, for example, in diagnostic microbiology and cancer therapy. The first step in producing a monoclonal antibody is to inject an animal with an antigen containing the specific epitope of interest. Each B cell produces a single type of antibody. B cells are isolated from the spleen and then mixed with myeloma cells, a type of cancer cell that grows continuously. Addition of polyethylene glycol causes the two types of cells to fuse together to form cells called hybridomas. The mixture of B cells, myeloma cells, and hybridomas are cultured under conditions which permit growth of only the hybridoma cells. Each hybridoma cell will produce a single type of antibody against a single epitope. The single hybridoma cells are then separated into individual wells of a microtiter plate and tested for ability to produce the desired antibody. The hybridoma cells producing the desired monoclonal antibody are then cultured. Monoclonal antibodies are isolated and purified.
Okay. So, cell-mediated immunity. Cells that are infected with intracellular microorganisms that have invaded and gotten through the outer coverings, the membranes, produce antigens or cause process of antigens that combine with a major histocompatibility complex, class 1 molecules, not the class 2s. Now we're talking about class 1 molecules. Okay. Cytotoxic T cells are stimulated to divide, and they produce more cytotoxic T cells. Cytotoxic T cells. What does cytotoxic mean? That means cell-killing T cells. Then you have the memory T cells as well. And when the MHC class 1/antigen complexes are presented to the T cell receptors, then you have cytokines that are released from helper T cells that also stimulate cytotoxic T cells. In cell-mediated immunity, okay, cytotoxic T cells lyse virus-infected cells that cause them to burst open and release those viruses. Tumor cells are also destroyed, and tissue transplants may also be attacked by cytotoxic T cells. Cytotoxic T cells produce cytokines and promote phagocytosis and inflammation as well. Okay, so let's look at the process. A dendritic cell acquires viruses by being infected or through the process of phagocytosis of infected cells. Viral antigens are then processed, and the processed antigen is bound to an MHC class 1 molecule. Okay, and then this may promote the antigen to form a naive cytotoxic T cell, and the CD8 then attaches to the MHC class 1 molecule. Okay, the beta 7s and the CD28s provide co-stimulation of the cytotoxic T cell, the B7 and the CD28. Okay, and then activated cytotoxic T cells produce interleukin-2 receptors, and it secretes interleukin-2. Okay, that interleukin-2 then binds interleukin-2 receptors. So what does interleukin bind? It binds interleukin-2 receptors. Sorry. Interleukin-2 then stimulates the cytotoxic T cells to divide and differentiate, and many divisions increase the number of T cells. And the activated cytotoxic T cells then give rise to memory T cells, which provide long-lasting immunity. The activated cytotoxic T cells give rise to functional cytotoxic T cells that kill cells harboring the virus. Okay. Helper T cells can assist cytotoxic T cell activation, and they recognize antigen presented by the dendritic cell. The helper T cells then stimulate increased B7 expression by the dendritic cell and secretes interleukin-2. Alright, so let's check it out. So activation of the naive cytotoxic T cells by antigens on the surface of the cell is activated. You got the naive cytotoxic T cell. Cytotoxic T cells then will split into two different pathways. It either is going to release cytokines and cause inflammation or phagocytosis, or you have the induction of apoptosis. Okay, apoptosis is where the cell just completely dies. Okay, it dies. And then that cytotoxic T cell would then move to another target cell. However, the naive cytotoxic T cells can then become, or should I say alternatively, could become memory T cells. And these memory T cells can form the secondary immune response.
When a virus infects a cell and synthesizes viral proteins, some of these proteins are degraded to peptide fragments. These peptide fragments are complexed with class 1 MHCs and displayed on the surface of the infected cell. Cytotoxic T cells interact with the virus-infected cells by recognizing both the viral antigen and the class I MHC. The cytotoxic T cell then releases cytotoxins which induce apoptosis in the infected cell and perforin which causes perforations in the cell membrane. Self-proteins are also degraded and the fragments presented on the surface via class I MHCs. This is not shown. Fragments of self-proteins are not recognized by cytotoxic T cells. Cytotoxic T cells remain intact, detach from the infected cell, and move on to other target cells.
I'm a virus, an infectious particle, an anathuga pirate, the genuine article, Ebola, chickenpox, West Nile, influenza, yellow fever, AIDS, herpes, also coming right at me. I'm not a cell, not an independent organism, don't even have my own metabolism. I only reproduce myself by taking over cells, then I bust them apart, no wonder you reveal well. The structure is simple, mostly proteins and genes, packaged as a sub-micron killing machine. A capsule's made of capsules and self-assembled. Whenever cells see me, it starts to tremble inside my capsid is where my genes reside. I got DNA or RNA on the inside, and the rest of me is various proteins. Its role is assisting in assaulting cells and taking control. I'm a virus, controls your cells around within. I'm a virus, your cells is alive within. The lytic cycle shows one method of attack. I attach to the self, surface piercing like attack. Inject my genes inside, use my victim machinery to make my genes and proteins to reproduce some more of me. Step 1, tail. 5 is grab the membrane on the wall. 2, inject my genes. The cell is gonna fall. 3, use the victim polymerase and ribosomes to synthesize my proteins, replicate my genome. 4, tail piece and capsule self-assembly. 5, I'm all together now. Look at all my progeny. 6 is lysis. I burst the cell apart. Now I'm looking for more victims I can really depart. I'm a virus. Controls your cells around within. I'm a virus. Sometimes instead of lysine, I misogenize, which happens after I inject my genes inside. I slip into your chromosome, deep inside your room. A menacing and silent presence, bringer of doom. Integrated in you, your DNA's my home. And see what happens when you replicate your chromosomes. Each copy that you copy copies me as well as you. You might make a million copies, there's nothing you can do. I lurk and hide in genes, call it prophy virus. And if you got another name, just call us a virus. We'll write it on papyrus till the moment I emerge from your DNA. I'm lytic now, a self-destroying surge. And suddenly your cells are dying. Watch me as I lies them. I'm a pirate, so forget you plan to program now, denies them. Your cells are virus factories and making millions more of me. I lose your cells and now I'm free, infecting you, my destiny. I'm a virus. Controls your cells around within. I'm a virus. Now meet my associate, the virus HIV, whose helpless target is a cell called T. HIV causes immunodeficiency, raising cancer and infection vulnerability. Remember that the T is an essential cell in the immune system army, it's the general. So knocking out the T cells sets the stage for cancer germs and viruses like me to invade. HIV's genes are made of RNA. Outside its capsids, a phospholipid membrane. The skin stolen from the cell at last dispatched. Studded with proteins that landed attached to a protein on the T cell, CD4. Inducing that cell to open up its door. Inviting in HIV enzymes and core. The T cells are already done for. HIV is a retrovirus, its sex routine has to do with transforming its RNA genes. Reverse transcriptase takes its RNA and reverse transcribes it into DNA. Then this DNA integrates into your chromosomes like a lysogenic linkage in your genome. This proviral DNA is transcribed and translated as new virus genes and proteins get created. And like any virus, all those parts self-assemble into new viruses, which of course resemble the original particle that started the infection, acquired by sex or a needle injection. I'm a virus. Controls your cells around within. I'm a virus. I'm a virus. Controls your cells around within.
All right. Hopefully you saw something really important there. Talking about helper T cells Okay, and T cells in general, and then towards the end of that video, the end of that song, you saw there is something that can completely wipe out your ability for your helper T cells or your T cells in general to function to help your body survive. Okay, yeah, the HIV infection, right? All right, let's move on. Okay, immune interactions. Innate immunity, antibody-mediated immunity, and cell-mediated immunity can function together to eliminate an antigen. So this right here puts several different things that we've talked about previously together. Okay, how helper T cells and dendritic cells are going to work together in the process to form naive cytotoxic T cells or naive B cells. Okay. Acquired immunity. Acquired adaptive immunity. Okay. How does it happen? Well, there's two different ways. There's active immunity and passive immunity. Active immunity is provided by the individual's own immune system. Okay. Naturally, antigens would be introduced through natural exposure. An artificial way to produce active immunity is when antigens are deliberately introduced in a vaccine. Passive immunity is when it's transferred from one person or an animal. Okay, from one person to another or an animal. Okay, antibodies from the mother are transferred to the child across the placenta or in milk. This is an example of passive immunity. Okay, naturally. An artificial way to develop passive immunity is when antibodies are produced by another person or an animal and are injected. Okay, so here we look at some system interactions with the lymphatic system in page 600 in your textbook. Please review this before your exam and your study materials and do your homework. Okay, it's only a two-part lecture this time. Hopefully everything is going great. If it's not, please message me and let me know what issues you're having. And remember, we can always meet anytime that you let me know ahead of time, okay, and discuss anything. All right. I will let you guys know on the Remind app everything, and I will send that out ASAP. You guys have a great week, great weekend, and good